Hello everyone, I am Shahan Layek and today we will learn about morphology of bacteria. So let us learn morphology of bacteria. First is cocci. Cocci means berry-sepid, spherical cells. There are several types like diplococci, streptococci. Diplococci means in pair, streptococci means strep means in chains. Clusters, staphylococci. And bacillus means rod-shaped bacteria. You can see several types of bacilli bacteria like coccobacilli, brucella, streptobacillus, okay, when bacilli are arranged in chains, streptobacillus, Chinese letter-shaped bacilli like cornybacterium, comma-shaped bacteria like Vibrio, Spirilla is spiral-shaped bacterium and that is Spirillum. Spirochete. Spirochetes are actually, chete means hair and spiro means coiled. So coiled bacteria with hairs. Actinomycetes. Actin means rays and mycetes means fungus. So these bacteria look like they resemble fungus and they have these actin-like rays. That is why actinomycetes. They have rigid cell wall. Another one is Microplasma. Microplasma means cell deficient bacteria. A bacteria that is cell deficient. Let us show you the pictures. First, cocci bacteria you can see. Diplococci which remains in pairs. Cocci bacteria, these are round bacteria which in chains. Streptococcus. And cocci bacteria in cluster. Staphylo. And you can see tetrads. Now you can see this one. These are bacilli, rod-shaped bacteria, coccobacilli, whose wide and the breadth are same. Now bacilli remains in chain when the rod-shaped bacteria are in chain. And you can see comma-shaped bacteria like Vibrio. And you can see Chinese letter pattern that can be found in case of cornybacteria. So these are the shape of bacteria. Now let us learn about the bacterial anatomy, the cell wall of bacteria. So we will study the two different type of cell wall which is there in gram-positive and gram-negative. So the first one is a gram-positive. This blue color is peptidoglycan and you can see in gram-positive bacteria the special thing that can be found is teichoic acid. You can see teichoic acid in gram-positive bacteria and this is the cytoplasmic membrane. So cytoplasmic membrane, peptidoglycan layer which is very thick in case of gram-positive and the teichoic acid in gram-positive. In gram-negative you can see the difference. The peptidoglycan has become very thin here. And we have lipopolysaccharide. The outermost layer is lipopolysaccharide in case of gram-negative. Lipopolysaccharide and lipoproteins. These are lipoproteins. So lipoproteins and lipopolysaccharide. Okay. Lipoprotein. These are lipoprotein and these are lipopolysaccharide. Between these two, lipoprotein and lipopolysaccharide, there is a outer membrane. Remember this, outer membrane. So its arrangement is just like this. First there is the cellular membrane, cytoplasmic membrane. Then we have the peptidoglycan. Then we have lipoproteins. Then we have the outer membrane. Then on the outside we have lipopolysaccharide. Okay. Now you can see the chemical structure of bacterial cell wall which contain peptidoglycan. And in peptidoglycan are actually made up of these two things, that is N-acetylmuramic acid and N-acetylglucosamine. So NAG and NAM, one is N-acetylglucosamine and N-acetylmuramic acid. And you can see tetrapeptide chain, pentapeptide cross linkage. So in this way bacterial cell wall is formed. Now let us see some of the picture how we can define bacteria. You can see this bacteria contains something which is known as mesosome. Let us discuss about mesosome. Mesosome are actually the invagination of the plasma membrane and it contains respiratory enzyme which function just like the mitochondria. Okay. And these bacterial cells do not contain mitochondria, do not contain endoplasmic reticulum. They have just DNA, but this DNA is not covered by nuclear membrane and nucleosome is not present. This is granular inclusion and these small particles are actually ribosomes. and the bacteria is always covered by a capsule. You can see this is the covering. Sometimes slime layers are also there. Okay, just remember this is the cytoplasmic membrane. After the cytoplasmic membrane, the outside layer is the cell wall, and this one is flagellum. Flagellum are actually the motile, helps in the motion of the bacteria, and these are pili. Pili are helps in the function of sex pili, and also they have the function of attachment, adhesion. The thing which is very important is the flagella. You can see the flagella. Let me show you the parts of the flagella. This is the flagella of bacteria, which consists of this part: basal body, hook, and filament. This is the filament, this is the hook, and this is the basal body. Now basal body consists of two parts, the inner rings and the outer rings. These two are outer rings, these are inner rings. Now inner rings is attached with cytoplasmic membrane, and outer rings, the two outer rings, the outer rings will attach with the outer membrane, and inner outer ring will attach with peptidoglycan layer. Okay, now let us learn about the arrangement and types of the flagella. So first one is the monotrichous. There is only single flagella. You can see example is Vibrio in Vibrio cholerae, and this one is amphitrichous. On both sides you have flagella. This example is alkali genes. Next is the Lophotrichous. Lophotrichous means on single side but a tuft or cluster of flagella is there. Example Spirilla. Next is Salmonella typhi. Next is Peritrichous, where the whole body is covered by this flagella, Peritrichous, and example is Salmonella typhi. Okay. Next is this one is Fimbriae, and I told you Fimbriae are hair-like appendages which helps in attachment. Okay. Now there are three types: common pili. This Fimbriae is also known as pili. Okay. Now common pili, sex pili, and call one pili. So remember these two, common pili and sex pili. Common pili are naturally there for attachment, and sex pili are for fertility, where the male insert the recipient female. You can see here through the genetic material is transferred with the help of this. The male transfer it to the female cell with the help of this pili. Okay, by the process of conjugation tube formation. Okay, and another thing which we will learn is bacterial spores. Spores are very resistant stage, and these are only formed when the environment is unfavorable. So this sporulation process is the process by which the spore is formed. Okay, and morphology of spore, let me show you with the help of picture so that you can understand how the spore looks. This is the spore. Sorry for it. You can see the structure of bacterial spore. This is the core of the spore, which is covered by a membrane, and that is known as spore membrane. This yellow colored part, the yellow part is the cortex, spore cortex. Next, the violet color, that is the spore coat, spore coat. So spore cortex, yellow color, and that is covered by this one, violet color, which is known as spore coat. And then this outermost structure of the spore, that is known as exosporium. Exosporium. Okay. Let us show you how this spore is formed by a cycle. Okay. You can see here. This is first the free endospore is released. From this free endospore will be germinate, and it will form vegetative cell. You can see the vegetative cell has been formed, which contain the DNA. Now this DNA will go replication. You can see DNA replication. Then the formation of spore, how it is occurring. One part goes to the end. One DNA goes to the end, and this form the fourth spore. And another, the majority remains with another DNA, and that is known as sporangium. Fourth spore, sporangium. So between that there a wall formation of sporangium and fourth spore. Then a wall formation occurs, and then the formation of double layer occurs. So within the bacteria you can see how the double layered sporangium which completely enclosed fourth spore. Sporangium has enclosed the fourth spore, and this has matured into endospore inside. So a bacteria that contains a endospore. Okay, and we discussed how the spore coat are there. There are different kinds of spore you can see here. Like you can see the types of bacterial spore. The first one is the central one. This is the subterminal, and this is the terminal. So all these are non-bulging spore. These are non-bulge, but these are bulging spore. You can see central spore, subterminal spore, terminal spore, and free spore. So this spore is free. Thank you for watching. Please subscribe our channel for getting more videos like this.